So, hello everyone. Yes, my name is Thomas Wolf. I'm a science fellow located in Frankfurt, and I would like to introduce Marc Streit. I'm very happy to announce him. He's a professor in Linz at the Johannes Kepler University and also one of the co-founders of Data Vision, which some of you already might from some really useful tools already being used in Bayer. And I'll just hand over to him to introduce himself a bit more and also talk about his work and ideas. So,
yeah, thank you very much, Thomas. It's a pleasure of to be here, and I'm going to talk about how to visualize insights from data. Let me see if that works. Yes. So what we do at my lab, the Visual Data Science Lab, is basically working on the intersection between those two worlds, right? We have AI and machine learning, and we have interactive visualization, which is sometimes the interface to the users. Not all the time, but it's usually some kind of human-machine interface. And you could do that, or you could look at this combination from two different points of view, right? You could use machine learning, which is here machine learning for this, for the purpose of visualization, which means you could let a machine learning model recommend visualizations, a dashboard to the user to work with to find insights, or you could do it here indicated as this for machine learning. You use visualization for the purpose of machine learning, which is also known as explainable AI. For instance, you have a model that learns something which is represented in the latent space, and you want to use visualization to understand what the model has learned. And we work on multiple topics in my group. So we do biological data visualization. Here you see a project together with Boehringer Ingelheim on patient cohort stratification. We work on provenance and reproducibility. So how do you track what users do when they make insights using visualizations, which is a bit difficult than doing reproducibility in Jupyter notebooks, for instance. We work on knowledge graph visualization, which is also an important topic in pharma. We work on onboarding, so how to onboard new users when they are approached with a new visualization or a new dashboard. Explainable AI, of course, and a topic that we work a lot with is embeddings. So, and this is what I'm going to touch upon most in my talk. And a couple of years ago, we teamed up with, or my team teamed up with two US labs and two friends, Niels Gelenborg at Harvard Medical School and Alex Lex at University of Utah. And we co-founded a company named Data Vision. And the idea was just to take the innovation from our research labs and bring it into production and bring it to industry. And why I'm mentioning this is because some of you might be using or might be familiar with a few tools that Data Vision developed over the years together with researchers and IT infrastructure personnel at Bayer. For instance, Elixir, it's a main it's an important tool for disease and target information retrieval, Patent 360, Literature 360, Hubble Search for accessing patent, literature information, clinical trial information, Marketplace for covering this ideation process before something is a project, like where it's still an idea, hidden for high throughput sequencing, and so on. But today I want to talk about what we do in my research lab. And some of you might be familiar with the idea of low-dimensional embeddings or projections. So the idea is you have a high-dimensional space, could be a document, could be an experiment you do that has lots of different dimensions, and you down project it to 2D space. So here, for instance, on the left, you see every point is a book, and you take all text of that book and you down project it where the similarity is represented in how close those two dots are in the scatter plot, right? And then you see clusters appear and you can try to interpret that. On the right-hand side, you see an embedding of a single-cell transcriptomics data set that appeared in, or that was described in the Nature Communications article. And the idea is again the same. You have a high-dimensional feature vector, basically, that you're down projecting in order to look at patterns in the data. The problem is that you really need to be careful when you're interpreting those down projections, because ultimately you lose a lot of information, right? It's just covered in two different dimensions. And of course, the hyperparameters matter that you use for running the algorithms. Cluster sizes can mean nothing. Distances between clusters can mean nothing. Random noise sometimes doesn't look random anymore, right? You see patterns, which is just noise in the data. So you really have to be careful. But I have to say, sometimes it's one of the few options we have, right? So we need to live with those limitations. A project that I want to talk a bit more is a collaboration with colleagues here at Bayer. I have highlighted the names of the collaborators. Thomas is among them. And it's work on the Chem-Informatics Model Explorer, short SIME. And the problem was that at that time, the machine learning group at Bayer was using a model that learns how to predict properties of molecules, right? Solubility, toxicity, drug likeness, and so on. So very useful. But the question is, based on what does the model make the prediction? What did the model learn? If you put in a new molecule, you get out the prediction of the solubility. And this is the prototype that is now also rolled out here internally. And I want to briefly tell you about the components. So you have this down projection, which is not more than a scatter plot. Every point is a molecule. Then you have a tabular overview of the chemical structure information and some more information that the model has learned. And a very important part is this attribution view. So you try to visualize what the model has learned that contributes positively to the prediction or negatively to the prediction, right? So in green, you would have parts of the chemical structure that contributed positively to this solubility, for instance, and then you also have contradicting parts of the chemical structure. And that allows then the domain scientists to really understand what contributes to the prediction and what has the model learned. Also by looking at neighborhoods of molecules that are close to each other. A follow-up project that we actually got published 2 days ago was this Bayesian reaction optimization, with Giulio Folpin and Thomas was also involved and many more. And the problem is a bit different here. It's so at that time, I think Giulio saw a publication that appeared in Nature, and it was a group from Yale that proposed Bayesian reaction optimization. So you have a chemical reaction, and the problem is that this chemical reaction has lots of different parameters that you can change as an expert doing the experiments, right? You have different ligands that you can use, different bases, different solvents, temperatures, concentrations, right? The question is how to combine those, how to pick those parameters in order to increase the yield, right? That's the optimization criteria. And based on this work, you also want to do explainability. So the question is, how does visualization can help? And this is the prototype we developed together in a kind of co-creation process between my team at JKU and Bayer. And what you see here is basically again at the top center, this embedding space. Now, every point that you see in red or orange is an actual experiment that has been done, right? And the idea of this Bayesian optimizer is that the model predicts the next X experiments to do, the next 5 experiments. Then you do that in the lab, you feed that back into the model, and the model makes 5 new predictions of next experiments to do. Of course, the experts, the scientists, the lab heads want to overrule that, being able to overrule that, but that's this wet lab in the loop process that is supported by AI now. And now these hexagons that you can see is what the model thinks about the space, right? So it tries to summarize what are the hotspots where you should go deeper, right? So here we pick this interesting triangular shape color map, which basically says blue is low yield, so don't go there. Yellow is high yield, so there's a good chance that the model will pick experiments there. And then if the brightness is decreased, it means that the model is not sure, right? You have certain spaces where the model says, well, I could do a prediction, but I'm not sure because maybe the samples that are used for training are not sufficient, right? And then you can try to understand, why did the model go into these yellow spaces? And as a human, you could also overrule this process, right? So that's the visual discovery tool that is then supported by this AI model. Another project that I want to talk briefly about is, and we saw in previous talks, it was already mentioning multimodal models, right? So you have a model that can deal with multiple modalities. It could be text, images, but it could also be others like videos, molecular structures, audio, right? And the idea of those models is that they put these multiple modalities in a joint space, right? It learns a latent space that can deal with those different modalities. The problem with that is because then it can make predictions, right? Because it knows, well, if you have a text, you predict an image that is in the close surrounding of this point. Now, there is something known as a modality gap. So there is a gap between the modalities in the latent space because those models are not perfect, right? This would be an extreme case where you see all image points and all text points, and they really are in separate spaces if you don't project it, which is a bad thing. But on the left-hand side, you see, for instance, a clip example, and you see then basically the model is picking the point that is closest to it. For instance, you see a user is typing simply simple sketch of a puppy, and then you pick the closest image to that point, right? Very simple, and you could do that sometimes in both directions. Now, we put that online as an interactive article because we have interactive visualizations there that tries to explain this modality gap, and then it's also looking at methods that are able to close this modality gap, or at least make it smaller, right? So it's not inventing new models that have a closer modality gap, but it's really about looking at existing models and trying to understand what are the downsides and how can this modality gap being optimized. And here are the visualizations that you can see. So on the left-hand side, you see a similarity matrix that basically maps every text point to every image, right? And you see then by looking at those lines in the scatter plot, the longer the line, basically the worse the match is, right? Because you want to have matches that are very close together, having no modality gap. And then you can interactively explore that. And this is made for machine learning experts. So in that case, it was the Machine Learning Institute at our university. It's run by Sepp Hochreiter, so the inventor of LSTMs. He's a very good collaborator, by the way. And the idea was that you can also plug in other modalities. For instance, they published a model that can do a prediction between, it basically takes molecules and can predict, it takes molecules and can, for instance, predict the microscopy image that you would get, right? So you have, you can switch those modalities, and that's a model that is optimized for those two modalities, microscopy image and molecule. And again, we use this approach to understand the modality gap in those models. Now, in the second part of my talk, I basically want to show you work that we did a couple of years ago, but there are some interesting updates. So some of you who saw my talk, I don't know, 4 years ago at one of the symposia, some of the slides, the intro slides are the same. So basically, the time curve technique is a technique where you have a time series data set, right? And you try to bend a curve by the similarity of the points along the curve. I will give you some examples because then it's much more obvious. So if you have, for instance, a camera image, a surveillance camera, every point is one image taken by the surveillance camera. The blob is basically camera images where nothing happens. If someone walks through the image, you see that this line is making a detour, right? Or if you look at different Wikipedia articles, if you look at abortion on the upper left, you see every point is a version of the same Wikipedia article. Time is along the line. And then you see there have been edit wars, right? Obviously, there are different opinions on abortion, and there have been big changes back and forth, right? This is what can be seen. And we used it to understand how Rubik's Cubes are solved. So who solved the Rubik's Cube before, or who tried? Who actually succeeded? Okay, congratulations. Now, there are actually humans that try to do that. The world record is 4.2 seconds, right? And machines that do that. But the question is, how can we explain what those algorithms do? And this was actually, it started with a bachelor's thesis, and that was the result. So you try to visualize what an algorithm is doing, and solving a Rubik's Cube is an algorithm, right? On the left-hand side, you have the beginner's method. This is what, if you would look up that on Wikipedia, you would end up with. And on the right-hand side is the Friedrich method, what is used in tournaments, for instance. So the idea is straightforward. You unfold this Rubik Cube virtually. You see it as a feature vector. You put points in the space and you connect it along the rotations, right? Every rotation will give you a new point. If you do that for the whole Rubik's Cube, you get a path. If you do that for many, you see patterns appear, right? Now, what do we do? What do we see here? If you look at cluster 3, the smaller dot is all solved Rubik's Cubes. This larger cloud above is almost solved Rubik's Cube. So you have just one piece that is missing. And then here in this cluster 4, you see that you need to do an awful lot of rotations in order to get from this larger blob to the final state. On the right-hand side, you see basically the same, but you see that you just need to go through this loop that is indicated as 2, which requires considerably less rotations. And this is then patterns that you can see. This was a setup for an open house day where you can put it in a Lego solver and it will be solved and you can watch it to being solved. If you give up, it was a Bluetooth tracked cube, so you can try it on your own first. And we developed a tool that allows exploration of such kind of data. But we applied it also to more serious use cases. So here you see, for instance, a tool that tracks what the user is doing. And then you project that and you try to understand what your users do with the tool, where they get stuck and things like that. So you can do serious stuff with that. I will. So here, this is, for instance, applied to chess openings. And this is the exploratory tool that we developed to see the different openings. And then you can like compare the different clusters and trying to characterize what makes one cluster different to the others and what did the professional chess players do. Yeah, here you see those openings, sucker tort, English opening, queen's pawn opening, and then we have some explainability on top of that. But the model also has problems. If you down project it, you see large gaps, although the player only made one turn, right? So there have been problems with this projection initially, and then in recent work, also with ETH Zurich, we tried to solve that and try to also come up with a strategy that not relies on the user to find all the patterns, but to find the patterns algorithmically and then just to explain them to the user, right? So that's the current work we are trying to do. And before I come to my end, I just wanted to mention like a recent project that that we now started with Boehringer Ingelheim, working on knowledge graph-based indication expansion. So a topic that is obviously important for probably every pharma company. The idea is you have a drug that like is connected to a certain target, and the target can then has a link to a disease, right? Which is then covered by a knowledge graph, right? So as an institution, you might have a knowledge graph that covers all that kind of connections from you and from your competitors' drugs. And then the idea is, how can we create a model, or in that case, our collaborators create a model that predicts that the drug also works for alternative diseases. And what we want to contribute using visualization in that case is that you have some kind of query interface, right? We heard about chatbots earlier, and you type in, for instance, as a user, obesity and some target, for instance, and then you get a small world view of, like, cut out of this whole knowledge graph that explains what you know about those entities. But then here in the sketch, this lower part is probably the most important one. You again want to use visualization to explain supporting and contradicting evidence, right? You want to look at other data sources that probably haven't been taken into account by the model to explain that a link predicted by the model is valid or not valid, right? And then again, the human has to decide. It's this human in the loop approach that we try to integrate. So I talked a lot about explainable AI, which is basically a combination of AI plus this plus a human. And I talked about these two different directions of doing research, but we can expect that new systems will do both. They will use machine learning for visualization and also do visualization for machine learning. And on the right-hand side, an interesting observation probably is that you can create very complex tools like the SIME and SIME for reaction that I was showing you. But of course, if you go down, so for model developers, they can probably work with those. Model users, it's already more difficult. Decision makers and then people who are affected by those tools, they need much more less complexity, right? So the solutions we provide need to be stripped in a way that the complexity is reduced. And with this, I'm already at the end and give the word back to Thomas. Thank you.